All right, thank you everybody for joining us for the DNA cell cycle analysis with Multicycle and FCS Express webinar today. Today we'll be discussing working with and modeling cell cycle data from flow and image cytometry data within FCS Express. For a little bit of background, Novo Software and Phoenix Flow Systems, the makers of Multicycle, we've both been in business for over 15 years and have partnered on many projects in the past. The Novo software team benefits from Phoenix Flow Systems' vast knowledge in the area of flow cytometry and their many industry relationships, while Phoenix Flow Systems has benefited from the ability of Novo software and FCS Express to quickly create custom professional solutions in the area of flow cytometry data analysis. Currently in FCS Express 5, we've collaborated to integrate the highly successful DNA cell cycle analysis application Multicycle. Cell cycle data works for imaging and flow cytometry data in FCS Express. The basic version of Multicycle is included for free with FCS Express. The basic version will work for normally distributed data that has one cycle. It does not take into account noise or background. The full version of Multicycle, which can be purchased as an add-on at the time of your initial purchase or after your initial purchase of FCS Express 5, works for multi-cycling data, and it will take into account noise or background information from your data. So what we're going to cover today is a basic introduction to cell cycle analysis, a little bit about the theory, how data is collected. We're going to be discussing some cell cycle artifacts, some things that you may need to take into account when you're working with flow or image cytometry data. And then we're going to get into working with cell cycle data using the Multicycle tool in FCS Express. We're going to show you how to work with auto-fitting of the data, how we can go back and manually fit some of the data, how we can change our overlays, how we can format plots to look nicer, and how to use all the tools that are available within FCS Express to get you from your data to your mathematically modeled cell cycle results. So let's start by discussing cell cycle analysis, some of the very kind of basic theory about how cell cycle analysis works, and how mathematical modeling of this data in FCS Express using the Multicycle DNA analysis tool works. So if you've ever worked with cell cycle data, you may be aware that as cells progress through the cell cycle, the amount of DNA in a cell changes from 2n to 4n. You can see this here that as we go through G0 to G1, and from G1 to S phase, there's a variable amount of DNA in the S phase, resulting in a 4n population in G2 and M. Again, this cell cycle repeats itself every time a cell divides. And what we can do with Multicycle and FCS Express is use these populations paired with fluorescent markers to find the 2n and 4n populations and everything that exists between that. So when DNA is stained using a variety of dyes, the fluorescent intensity is going to be directly proportional to the amount of DNA in the cell. This is going to allow you to define those G1 and G2 peaks, again, as well as that variable amount of space in the S phase. And when you look at the theoretical model of DNA analysis, kind of over here on this screen, you can see that there's a discrete 2n population and a discrete 4n population. And mathematical modeling of this data takes into account many aspects of this theoretical model that are not as simple as it's displayed on this little graphic. The theoretical modeling includes the distance between the 2n and 4n peaks. So you should actually have a ratio here of 2. You should have double the amount of 4n as you have in 2m. Mathematical modeling is also going to take into account the CV, or the spread, of the G1 and G2 peaks. But what happens in reality, rather than what you see in this figure, is that in biological samples, the CVs of the peaks are wider than one bin, of course. And you can see here with some real data, you can see our G1 peak is actually much wider than one simple line, and our G2 peak here is actually much wider than one simple line as well. The other thing that can happen with biological data is that the 2n to 4n ratio might be significantly or slightly greater or less than 2, the theoretical derived value. And you can also end up with debris, noise, background, or artifacts within the data. So let's talk briefly about artifacts for working with cell cycle data, and some things that can happen with your data, some things that the multi-cycle modeling can take into account, and also some things that you can do in FCS Express or working with your cell cycle data to kind of clean things up, so to say, a bit. And one common artifact that occurs is because cells, when they go through a flow cytometer, they don't always pass a laser in your flow cytometer one at a time. There isn't this kind of magic focusing in there that's taking place all the time. Sometimes you can see here when we have a laser and we have cells going through it, 2 cells will stick together. And when 2 cells or multiple cells, it doesn't have to be 2, it can be 3 or 4, or actually large clumps of cells pass the laser, they'll give you populations that are greater than 4n. So if 2 G2 cells stick together, this may look like an 8n population. Or if 2 G1 cells stick together, this may look like a 4n population. Or if a G1 and a G2 cell stick together, they're going to fall somewhere between as an S phase population. So all of these things can happen. There's many combinations that can occur from cells sticking together, of course, but generally these are referred to as doublets. And in most cell cycle analysis experiments, you should be collecting width, area, and height parameters, which will help you tease out single cells from doublets. And you can see here with this particular experiment, we've done a few things. We've acquired our side scatter versus forward scatter parameter. And we've also acquired our FL2, which is our DNA cell cycle parameter, width and area parameters, as well as our height and area parameters. So by using a combination of these gates, you can see here that with our data analysis, we have this 4n plus kind of situation, as well as some kind of things going on down here lower than 2n. When we apply our scatter gate to that particular multi-cycle plot, you can see that this actually starts getting cleaned up a little bit. We're losing some of that kind of 4n plus 4n populations up here, as well as cleaning up some of the things around the debris area or things that are less than 2n. And if we take this a little bit further, and we have our area versus height parameter, or area versus height gate, which is one of our singlets gates or doublet exclusion gates, as well as our area versus width gate, when we apply this gate to the plot, you'll now see that almost all of this 4n plus population, anything kind of to the right of this blue G2 peak, has disappeared. And again, what we did there was just exclude doublets from our analysis. Doublets are anything that's more than 2 or 3 or 4 or a whole bunch of cells stuck together. We do have this population down here on the left, though, that's less than 2n. If we make our doublets gates or double exclusion gates a child of the scatter gate, that gets further reduced. But the benefit of using the full version of multi-cycle as well is that any of that noise or kind of background that's included here will be accounted for with your cell cycle modeling. So they will not be included in your results when you're calculating your G1, G2, and S phases. So again, we're going to talk about working with those background parameters a little later as well. But you can always come in and you can set some of these exclusion gates, acquire your data correctly to get some of this. And one of the other kind of things you always have to discuss when you're working with cell cycle data is the kind of premise of garbage in, garbage out, right? You need to acquire your samples carefully with the correct parameters. And if you have good data coming into your cell cycle model, you should have no problems modeling that data. Now, with image cytometry, we have some image cytometry data here on the right, and we have the individual cells that are associated with all the bins in this histogram here in what we call a data grid. You can have some other artifacts occurring. And they're actually very similar artifacts. And in segmentation routines, which define where a cell is in your analysis, segmentation routines can mark 2 cells as one. They can cut off a piece of a cell. Cells can be overexposed by your microscope. All of these things must be taken into account. And what we've done here is we've set a marker gate to give you a little bit of an example of what can happen here. When we apply this image cytometry marker gate to our images, we can see that when we look at cells that are kind of falling on the low end here, there's some blanks. There really isn't anything going on there. These might be just segmentation artifacts where the computer thought a cell was there, although nothing existed. We have another case here where a piece of a cell is actually being cut off. Another one here where a piece of cell is being cut off. So these are probably things that you'd want to exclude from your DNA analysis if you're working with image cytometry data. Now, conversely, if you look on the higher end here, you can see that many of these cells are actually overexposed. So we'd want to exclude them from the analysis because we don't have that directly proportional link between the fluorescence data and the DNA content of the cell because they've just been overexposed. The other thing that you can see in some of these is that in some cases, multiple cells were collected here as part of the segmentation routine. In this case, 3 cells were collected. So these are all things that you may want to exclude from your analysis before you begin. And again, you can do this at a kind of basic level by creating a marker gate or creating some sort of gate on your image cytometry data and say, let's only look at the data that falls within this particular section. And when we do this, we can see that we have nice kind of single cells that aren't overexposed or underexposed, which we can be using for our cell cycle analysis down the line. So again, artifacts are one thing that you should be thinking about and taking into account when you're working with this data within FCS Express and using mathematical modeling. So with that being said, we're going to actually go into working with Multicycle in FCS Express. We're going to show you kind of how easy it is to do and some of the things that we can do within Multicycle. So to do so, I'm going to bring up a layout that I've actually started creating. Again, this is a little bit of a more advanced webinar. We're not going to show you how to actually insert standard plots and create gates and gating hierarchies. But the idea with this data is that we've set up a scatter gate, we've set up a doublet exclusion gate, and our fluorescent parameter to be using here is our BL2 parameter. So I'm going to create a new page and show you how to insert a multicycle DNA analysis plot. Very simply, you click on the Insert tab, you choose Multicycle from our 1D plot selections, and you draw an object on the page. When you draw an object on the page for your multicycle DNA plot, FCS Express is going to prompt you to choose a parameter to use for your DNA analysis. In this case, BL2A is the DNA fluorescent stain, or the parameter where that stain was collected, so we will click OK. So a multicycle DNA analysis plot has now been inserted into our layout. If we now click on the multicycle tab, you can see that it's become active. It says that we're auto-fitting the data because this is highlighted in yellow. We're auto-fitting to use model 1 for this particular data. And on this plot, it's more than just a histogram at this point. You can notice some kind of blue hatch marks. You can notice that there's a G1, an S phase, and a G2, as well as a green line kind of fitting over this data. So let's first talk about these different overlays or these different displays on this plot. If we right-click on the plot and choose Format, this is where you can access all of your formatting information, really for any plot in FCS Express. But if we come into the overlays of the multicycle plot, you notice that we have our different overlays indicated here. So our noise parameter or our noise fit is going to be this hatch blue mark, which you can see down here on the left. Multicycle is determining what is going to be noise in this assay. The fit is being determined as a green line, which is the green line that we've defined here. And of course, our diploid G1, S, and G2 phases. Now, one nice thing you can do in FCS Express, again, this kind of comes into reporting of the data. We don't have to use the defaults that we use here, and you can even change your defaults so that every time you open up a plot, the way that you want to view this data is happening. But generally what I like to do with my multicycle plots is I change the fill style to something that's just full rather than a hatch mark. I'll do that here. I'll change some transparencies. I'm also going to change the color here for my different phases. And in the end, we'll show you what this kind of looks like. Let me change to green. Now when I click preview, you can see that I've formatted this plot to look a little bit nicer. My G1 is located in this red transparency, my S phase is this blue transparency, and my G2 is this green. So again, these are more aesthetics for working with the multi-cycle plots. It is also kind of useful to be able to visualize these in a way that makes sense for you. And we also do want to explain where these different overlays are occurring. So once you have that plot inserted and you have it formatted however it is that you want to look at that, what we can do is actually start deriving some statistics for this multi-cycle plot. So if I right-click on a plot and I choose statistics, rather than a regular histogram, we now have the ability to choose DNA cell cycle statistics, DNA experiment statistics, and DNA model summary statistics. So I'm going to insert all three of these. I'll start with the DNA cell cycle statistics. I'll then follow up here, and I'm going to choose my DNA experiment statistics. And I'm going to finally put in here our DNA model summary statistics. So again, these are the default statistics that are provided by Multicycle. What the DNA cell cycle experiment or DNA cell cycle statistics show you are in this box, and they're kind of the default things that you would expect for working with cell cycle data. You're going to have the value for your G1 and G2 means. You're going to have your percentages for G1, S, and G2, as well as the ratio that was used here. So again, this is the G2 to G1 ratio. In theory, this should be 2. Our mathematical model is determining it's a little under 2, which isn't so bad. So these statistics will be here as you navigate between different data files that you've put in your data list. They, of course, will update for you in real time. Now, our statistics here for DNA experiment statistics are located here on the bottom left. This is essentially an interpretation of the data by Multicycle. And you can see here it says that this is a multi-cycle suggestion. This is just a guideline only. So this is a basic look from a software perspective about what's going on with your data. It's saying that there's no abnormal DNA content in here. There is diploid percentages here, and here's our S phase confidence. And in this case, with this particular model, it's pretty good. We also have some values for the experiment statistics, the chi-squared value for the model that we're using, as well as the number of cycles and the cell cycle fits. So again, this is a very kind of general way to look at your analysis and to kind of determine if things are okay. I mean, if this comes up and it says your S-phase confidence is very bad or it's very poor, you're driving some statistics that don't seem to make sense. Of course, we want to come in and maybe change that model or have a look at how these things work. Now, when we're working with our DNA model summary statistics, which are located in this table, what this table is referencing is each one of these models is one of the models to display that's located in your multi-cycle tab here. So there's 6 different models. They're listed in order here. SL S0 is model 1, going down to our CV's fixed, which is model 6. And as we change between our different models here, using the multi-cycle tab, we can see how these models are adjusting on the data analysis itself. With these model summary statistics, one of the values that a lot of people like to use to make a determination whether a model is a good fit or not is the chi-squared value, where generally a lower chi-squared value is going to indicate a better goodness of fit. So when we click on model 2, because it has a 1.87, we can see where G2, G1, and S phases are with that. If we go over here to our aggregates model, which has a value of 2, we can see how that changes. As well, again, with our different models. So you may have to pick and choose, depending on your data, which models you want to use. Of course, if you're working with some data and you have chi-squared values, that are very, very high, with some other ones in there that are very low, you may want to go through there and kind of review which one is going to make sense the most for your data. And again, the model can only do so much, right? We are going to model the data. We're going to try and use Multicycle to do it as quickly and as efficiently as possible. but you do want to go through and review that data carefully and make sure that it makes sense. Now, along with the different model types, there are different cycles that can be analyzed in our cycle tab here. You can see when you're actually using the full version of Multicycle like we're using now, you'll have access to more than just the one cycle that comes for free with the software. You'll be able to come through and look at overlay peaks, try and calculate an apoptotic peak if one exists, aneuploids, 2-cycle, 3-cycle, as well as synchronous S phases. And to kind of show you how that works in FCS Express, I have a series of data that I've opened up here in our data list, and I'm going to insert a plot, a multi-cycle DNA plot for this data, and we'll see what multi-cycle does. So I'll expand this plot type a little bit. I'm going to show our DNA cell cycle statistics. And you can see that with this particular plot and with this particular data file, Multicycle has determined that a 2-cycle fit is best for this data. There is an aneuploid population in here that's determining. You can see that we've located our G1 and our G2 means for both the diploid and aneuploid populations. And if you're working in the full version of Multicycle and you have these kind of population and these changes between data files, as you navigate through the data list, Multicycle is going to automatically try and fit the data to these models using the cycle that makes the most sense. So as I continue on here through our data list, we can kind of see, again, these are a lot of 2-cycle models. We get into something a little bit more complex where we've actually determined 2 aneuploid populations as well as a diploid population. But Multicycle is going to do its best to kind of tease these things out for you. And if you're working with more than 1 cycle worth of data, using the advanced Multicycle, if you're working with aneuploid populations, it's going to be a great benefit to you as well. So coming back to our standard kind of 1-cycle population, I want to start talking about what happens in the cases where you need to start doing some manual adjustments to this data. Maybe where the autofit isn't quite fitting exactly as you had planned. Maybe there's some kind of variability within your data that you need to adjust for. That's when we can actually turn off this autofit function. And when we turn off the autofit function, we'll have the ability to come in and change our models. So we can compare different models. We can change what's being included with the background and some of our fit parameters. And instead of using our Multicycle tab here, I generally like to show or also use, when we right-click on a plot, we can choose Format. And in this formatting tab, you notice that there's actually a few more things that you can choose for within the DNA section. This is a special section that's going to appear with your cell cycle data only. This is going to allow you to have all of the general options that we have up here, access to the fit cycle parameters, our background parameters, as well as showing different position markers on the data. Now, I'm going to start with position markers. I find this a very useful tool in Multicycle. What we can actually do is say, if we want to show where the G1 mean is, and if we'd like to show where the G2 mean is, you can show this position marker on the plot. I'm also going to make my position marker for the G2 mean be green, and I'll up the size of these a little bit so it's a little bit easier to see for everybody at home, and click Preview. And you can notice now on our Multicycle plot, we've shown and we have these little bars here for where our peak mean values for the G1 and G2 are. I'm going to choose them to display for now, and we're going to come back to them and show you how you can actually use them to start changing some things about how this data is being modeled. But coming back here to our general tab, just to give you a little bit more background about what this actually means. Again, this is the fit model to show, so this is really just a duplicate of what you're looking at up here in your model to display. So if you wanted to change this from the formatting tab, you can simply pick and choose which fit model to show. We also have the option when you work with this multi-cycle plot, or you work with multi-cycle plots in general, you can pick and choose which parameter you want to use by default. In this case, in our default situation is to prompt you whenever you put in a multi-cycle plot which parameter to use, but you can also just say use the parameter below, and you can match up the parameter number with whatever parameter number your fluorescent value or your fluorescent dye for measuring DNA content is. Now, the cycle tab or the cycle category is really where you can come in and change a lot of the fit parameters for your data. This is a very important and very kind of powerful tool to be aware of. Besides just changing our fit types, which we can do from here, if we wanted to take this off of the automatic mode and we wanted to see what a 2-cycle fit looks like for our data, we can simply choose 2-cycle and click preview. And we can see what a 2-cycle fit would look like for that data. And of course, in this case, 2-cycle fit isn't appropriate. The 1-cycle fit that was determined would be the best. But maybe you have a case where you have a 1-cycle fit and you have some data with apoptosis peaks, right? This is a great place that you can come in. You can say, let's see what a 1-cycle peak looks like. And then let's also come in and see what a 1-cycle peak with an apoptotic overlay looks like. So you can change that. You can pick and choose. You can have multiple multi-cycle plots within FCS Express. You're not just limited to having this one plot. You can, if you want to look at 1 cycle in one plot, and you want to look at 1 cycle with a apoptotic overlay in another plot, you could do that on the same page to kind of get an idea of what makes sense for you to use. Now, in the fit model properties, another property that you can change is the estimated diploid CV value here. And you can see here that in our multi-cycle statistics, we have our G1 CV values, or the spread of G1, and our G2 CV values, or again, the spread of the G2. If you wanted to constrain this value or you wanted to expand it, for whatever reason, maybe you've acquired some data that has a very narrow peak, maybe it has a very broad peak, and you want to change that around to see how that will affect your model, you can simply type in a new value here to kind of expand that to a certain degree. If you type in a value that's kind of too high, if you type in a CV value of 100, of course, that's not going to work because, modeling the data using that value is not going to be correct. But there's an option here to also set the G2 and G1 CV values the same. So if I choose this option, it's going to say that for our data, we're going to assume that the G1 and G2 CVs, they have the same spread. This may or may not be of use to you in a certain case, but it is here for you to define if you'd like to change that. Now, another important thing that multicycle needs is the G1 and G2 means. We need to kind of know where the peak of your data is in each of these populations. So when we auto-fit the data, or when you bring in data, FCS Express and Multicycle are going to make their best guess about where your G1 and G2 means are. And you can see here we've actually made a pretty good approximation of where those are. But if your data shifts, if it's moving around a little bit, if Multicycle isn't adequately determining where these values are, you can simply come in here and change these values. So if I wanted to manually input a value of 2 million here, we're automatically going to try and use that double the G1 value. We click preview. We can see how that changes on our plot. And we can see that we've actually moved this G1 mean because we inputted 2 million. We've slid down our G2 mean, and we've also adjusted for our G2 G1 ratio that Multicycle is determining to be the best fit for this. So again, you can come in here and you can change these values if you'd like. And the one thing I should stress with this, the more that you change values within this multi-cycle formatting tab, it really means the more that you're getting away from the theoretical modeling of your data. So if you have to come in here and you have to make changes for each of these sections to kind of make something fit or make something kind of work with your data, you may want to go back and have a look at that data and make sure that what you collected has been collected appropriately and that it can be appropriately kind of fitted for that data with Multicycle. So again, some minor changes I think are okay, but if you're coming in here and you have to, your means are 3 times apart and they're very, very different, and your G1 G2 ratio is 5, you want to rethink that data, right? But mentioning that, the other option that we have here is to change our G2 to G1 ratio. You can see with this particular data set, it's 1.9 as opposed to the theoretical value of 2. So in this case, it makes a lot of sense with this data. Our 4N populations are twice as much as our 2N populations. But you may run into cases where this value needs to be changed and you need to use the fixed value. So you check this box and you would manually type in a value. So what I'll do is I'll show you some data where this has actually occurred and where it actually makes some sort of sense to change that value. In this case, we're looking at some image cytometry data, and you can clearly see there's a G1 peak, there's an S phase, and a G2, and we fitted that data correctly. But this is because we've unchecked the auto fit, and what we did with this particular data set was we came into the cycle and we said use a value of 1.6. If we were to come in here and turn our auto fit back on, you can see that when we auto fit the data, this is what Multicycle determines as its best fit for this data. And of course, it looks like this does not make sense for this particular data set. And for whatever reason, when somebody had imaged this data, the G1 to G2 values, even though they exist, and we can kind of see that in the histogram, they were closer together than the model would expect. So again, the way that that's changed is that you would come in here to your formatting tab, and instead of using the value of somewhere around 2, which is what Multicycle would determine, you would come in, you would enter a fixed value here, and we would check that box, use 1.6, and now our data is being fitted appropriately to what the data looks like. We're still taking into account a reasonable CV. We still know it's one cycle data. We have our G1 and G2 means, which look acceptable, and we actually review that with the data. But we just know with this particular data set, the spread between these G1 and G2 populations wasn't as much as would be expected. So again, that's a change that you can come in and make if you'd like to use that as well. Now, the other option that we have is in that cycle tab is for the order of S. And what the order of S means here is actually the shape that the S phase model is being determined for. So by default and by automatically, we will be using an order of S of 0. There's actually 3 orders of S that you can choose from, 0, 1, or 2. And the 0 order of S assumes that the S phase is in some sort of rectangular or broadened rectangular configuration, or that's how it's kind of appearing with the data. If we were to come in and change this to an S order of 1, what this is assuming is that the S phase is some sort of kind of broadened trapezoid that's tilted in one direction or another. So when I click preview, you can see here that when we use an order of S of 1, we have this kind of decreasing S phase. And for this data set, it doesn't necessarily make the most sense, but if you have a decreasing or increasing S phase as it progresses from G1 to G2, an order of S of 1 might be useful. And in the kind of other extreme case here, you can use an S order of 2, which generally means that you're going to have a kind of tilted S phase, but it's assuming that on the sides, as you're closer to G1 and G2, that the S phase is a little bit higher in that account. And with this particular data set, we don't see much difference between the order of S when we're looking at 1 or 2, but there is, of course, a great difference when we're looking at an order of S between 0 and 1 or 0 and 2. The other thing you'll notice is that with order of S, we do have a model here where we can set the S order to 1. So besides coming in there and kind of changing these parameters manually, if you do have a decreasing or increasing S phase, you can simply choose model 5. And model 5 is going to kind of take that into account without you having to manually come in and make some of those changes with the data. Now, the other options that we have in here are essentially for the background modeling. And you can see here, again, we have the background or the noise for this data highlighted in a blue kind of hatching. And if we right-click and choose Format, we have an entire section here for background fitting. This is all, again, provided by Multicycle with the advanced version. This isn't in the free version. But if we uncheck use background fit, you're going to notice two things. One is I want you to watch as this G1 value changes and also as this background modeling disappears. You can see our G1 went from 35% to 33%. Our background modeling is removed from there when we uncheck use that background fit. So, if you do have background data and you've kind of decided not to use either doublet discrimination or you're kind of including a little bit more in your scatter gate than you would have expected, using the background fit is a great way to kind of correct for some of these things. You notice that the background doesn't just subtract from what's prior to the G1 peak, where it's doing some subtractions all the way across S phase and G2. So it's a very sophisticated way of analyzing this data. It uses some kind of clumping algorithms. You can do corrections for the diameter of what's used in that background fit, as well as using a sliced or unsliced nuclei way to fit that background. And all of this information about background fitting, we could talk for a very long time about this, but it's all outlined on the Novo software website. I'm going to give you the information about where you can find, again, what these different models can do and how the background fitting options can be applied to your data. But I think it's just important in general to know that these exist here, and that if you want to use background fitting or if you determine that you don't want to use it, you can simply come in here and uncheck it. Again, you can also access this through the Multicycle tab through the background button right here. But this all kind of brings us back around to the position markers. The position markers that I've defined here on the plot for the G2 and the G1 phases, again, they're defining where the peaks for those G2 means are that are in there. And instead of actually having to go back and forth through our formatting, if you know you need to shift either your G1 or your G2 peak a little bit to kind of meet or kind of reconcile where the data is on your plot to what the multi-cycle fit had been determining. The multi-cycle, these fit markers are really great because you can actually move them directly on the plot. So when we have one defined, you can see that you can actually shift that left or right, whether it be the G1 or the G2. And multi-cycle, when you make these changes, is going to try and recalculate the model based on where you had placed these position markers. And when it makes those changes, when we come into that formatting tab, you'll notice here that in our cycle, it did actually make that change there. So instead of about 2.4 million to 4.8 million for G1 and G2 means, you can now see our G2 mean is set up around 5.2 million. So again, this is a kind of quick and easy way that you can come in if you're working with a kind of a batch process with data and you're noticing that some of your G1 or G2 means are shifting around a little bit for one reason or another. You can kind of make a quick adjustment on the fly by adjusting these markers on the data. Now, once you kind of have all of this together, you can do quite a few things. You can change this kind of formatting any way you like to kind of come into having this really kind of nice report like what we're showing here on this page. Another kind of way that you can work with this data in FCS Express 5 is to set up pie charts in the software. I encourage you to kind of take a look at our website and our other recorded webinars about how to create these pie charts. But the great thing about this, again, is as I move a gate, our model will be recalculated on the fly. It will adjust wherever we have that data displayed, whether it be in some set of pie chart or within an analysis template ourselves. You can also notice that when we look at this plot versus this plot, we've actually zoomed in on the axes here a little bit. When we're actually looking at some of these lower frequency aneuploid populations, or maybe even your diploid is a lower frequency than your aneuploid, you can change the formatting on these axes. You can change anything about these plots that you like, but if you right-click on your multi-cycle plot and choose Format, a very common thing that people do, again, is zooming in. You can simply come in here and in your y-axis values, you can change to a new kind of maximum. So if we wanted to zoom in from 0 to 150, we can change that value, and we get a little bit better view now of kind of what's going on behind the scenes, especially here you can see with our background modeling, and if we had other populations that were overlapping, it's a very kind of quick and easy way to do that. Now, one of the final things I wanted to discuss about working with multi-cycle plots is the ability to create a gate directly on a multi-cycle plot itself. And the reason you might want to do that, I will just actually grab our scatter plot page, plot from the first page, is if you wanted to compare say your G2 to G1 on a different parameter. So we've defined our DNA analysis and our mathematical modeling based on our BL2A parameter, but we also have a BL3A parameter. And we want to look at how G1 and G2 compare with this parameter for these different populations. So what we can simply do in Multicycle or in FCS Express is we can create a marker on this multi-cycle plot. And I can convert this marker to a gate by right-clicking and choosing Convert Marker to Gate. We'll choose Convert and Link, and we will call this G2 marker, which has appeared on our second page. And now we have a G2 marker gate defined. And again, if we come in here and we create this, kind of based on where our modeling is, we can convert this marker to gate, marker 2, and we'll call this our G1 marker. And what we can then do is with our gate view or with our gating tab here, you notice that we do have our G1 and G2 marker gates. So if I wanted to apply G1 to our plot on the top and G2 and our plot on the bottom, we can now get an idea of the frequency and the distribution of our BL3 marker as it applies to our G1 and G2 populations. Again, we're not accounting for the overlap between S phase and G2 with our markers. That's all being defined by the modeling. But if you do need to kind of get into the data a little bit further, you can use the breaks from our modeling here to kind of set where these G1 and G2 markers are going to be and apply that to any other plots that you'd like to in the analysis. Now, the other thing that we can do in FCS Express in relation to multi-cycle plots is a lot of times, if you have been working with histograms and maybe you didn't know that multi-cycle existed, maybe you've just purchased the full version of multi-cycle, or you just wanted to compare a histogram to a multi-cycle plot. If you have a histogram already created in FCS Express, you can simply select that histogram, come into the Formatting tab, and change the plot type to a multi-cycle DNA analysis plot. When we do so, FCS Express is going to convert that plot to a multi-cycle plot. We'll now no longer be using these G2, S, and G1 markers that we've manually defined because we have a more sophisticated approach for analyzing this data. And you can simply come in here and start grabbing your multi-cycle DNA statistics, and you'll be kind of off and running with your histogram now that you've converted to a multi-cycle DNA analysis plot. And again, you don't just have to use one or the other. If I do a few undo steps here, we get back to our histogram. If we really wanted to kind of compare these things side by side, we can simply put our multi-cycle plot over here on the right, or histogram on the right will change to our multicycle DNA plot. And you can see here that here's our multicycle DNA plot. And as I previously mentioned, with your multicycle plots, you don't have to use just one at a time. If I wanted to compare in my top plot here, model 1, and kind of leave that as a baseline, and on the bottom I wanted to start looking at model 2, you can actually just select this plot on the bottom, start changing your models around for the different plot type that you've inserted, and see how the differences apply between different models for your data. So again, you can work with as many different multi-cycle plots that you'd like to work with at one time for one or multiple data samples within FCS Express. So that covers many of the kind of basics to even some of the more advanced features of multi-cycle within FCS Express. And we did want to kind of end today with just kind of a basic summary about some of the things that we talked about. The basic cell cycle analysis with the multi-cycle plugin comes for free with FCS Express Flow and FCS Express Image Cytometry. Again, what this is going to allow you to do is model your data for one cycle, not including background or noise. And if you want to get a little bit more advanced, if you have multi-cycling data, or if you need to be using the background and noise and aggregate adjustments, you do want to have the full version of multi-cycle. All of the plotting, the data analysis, the mathematical modeling, the advanced report generation tools, when you're using multi-cycle in FCS Express, they're all together. That's one of the really great things is you can have those pie charts deriving directly from your mathematically modeled data. You can have bar plots, regression fitting. You can have all of this included in one package in one software, so that way you're not having to move back and forth between different things. Now, FCS Express Image Cytometry uses all of these tools that are kind of proven tools for flow cytometry analysis, including the multi-cycle package, to improve what you're doing with your image cytometry data. So if you're kind of manually setting markers and gates on your G1, S phase, and G2 populations now with your image cytometry data, you can take this kind of a step further and get a little bit more sophisticated, account for the overlapping populations from G1 to S and S to G2, as well as with the advanced version, take into account any of that kind of background and noise. Now, again, we also provide many resources for our customers, such as this webinar. We have many tutorials, sample data, online forums, and customer support, so you can get up and running with your data and your multi-cycle data very quickly and easily. I do want to show you on our website. If you come to our website and you just type in multi-cycle in our search tab here, you're going to be directed to many of the resources that we have for working with multi-cycle and FCS Express. I really like this kind of first page that we have here about working with the multi-cycle plugin. It tells you a lot more about multi-cycle itself, how the fitting engine works, and there's also links to our multi-cycle cell cycle data display explained. So I hate to say DNA analysis for dummies, because nobody in this room is a dummy, of course, but this really kind of breaks down what all the cell cycle statistics mean. It breaks down what the model summary statistics mean, what these different models do, what the background modeling does. So I really do recommend coming to our website, looking for this cell cycle data analysis explained document. Again, in our manual, we'll have all of this information. Again, you can just type in multi-cycle on our webpage and get to this location. It's going to tell you more about how all of these different models are used and what they actually mean. And at the end of the day, once this webinar is completed, this is where the recorded version of this will live as well. So thanks again for joining us today. We're going to be opening up the floor for some questions here as well. I see a few have come in as we were working today. And we hope you enjoy working with FCS Express and Multicycle.